Hello, I'm Dr. Greg Maguire. Today I wanted to share with you some information about stem cells, particularly adult stem cells and adult stem cell derived technologies that have been used to make therapeutics, particularly to develop therapeutics for skincare. So to do this, I want to share with you some slides to better tell our story. I'm going to do this, bring up the slides here. So I'll be talking about human stem cell release molecules and how these molecules derived from stem cells are used to treat compromised skin. As a brief background to me, I'm a former UCSD medical school professor. I was awarded the Fulbright Fogarty Fellowship from the National Institutes of Health here in the United States. I have many peer-reviewed publications over the years. I continue to do that work. Just a couple of months ago, we published a paper, for example, with the Physiological Society about how adult stem cell release molecules treat neurodegenerative diseases. Last year, I released my first book. I'm working on my second, which will be published later this year. The first book was entitled Adult Stem Cell Release Molecules. I have a number of patents, and again, I continue the research on adult stem cell release molecules and how they treat not only brain conditions, but of course, skin conditions. That work is continuing. So let's first talk about what are stem cells. Stem cells really are, you could think of them as the raw material for building the body. They are those building blocks that create all that you can see, but also all that you cannot see unless you use very specialized technologies to be able to view these cells in action. And I wanted to let you know that not all stem cells are created equally. There really are a number of different types of stem cells. And as we look through the different tissues in the body, stem cell types, there's some basic categories, but there are very nuanced differences between the stem cell types. You'll forgive me today, I'm dealing with a respiratory tract infection, so I may be coughing a little bit and sipping my tea here. So please pardon me for that. But let's talk about categorizing stem cells into two basic types, embryonic versus adult stem cells. So those embryonic stem cells that you've heard much about over the last couple of decades, those embryonic stem cells come from embryos. So the fertilized egg, so the egg fertilized by the sperm, about 3, 4, 5 days following that, develops embryonic stem cells. And these embryonic stem cells are what we call totipotent. That means those embryonic stem cells can create pretty much any cell type in the body, including creating adult stem cells. There are a number of ethical issues using embryonic stem cells. They became quite controversial, but more importantly, they're difficult to deal with. They've been shown to easily turn into a cancerous cell type, and equally important, using embryonic stem cells for therapeutics really doesn't offer the efficacy that adult stem cells do for most conditions we look at in human beings. So adult stem cells, the preferred stem cell type for therapeutic development, they're found in most, if not all, of our adult tissues. And in each of our tissues, different types of adult stem cells are there for maintenance and healing of that tissue. So we're constantly working, doing things, needing to recreate the physiology of the body, and the stem cells are there for the recreation the daily recreation of all that physiology that needs to be maintained. Furthermore, if a particular tissue is injured, whether it's a skin injury or an injury to the brain or to the arteries, stem cells can come in and help to heal that tissue. Also, with adult stem cells, unlike the embryonic stem cells, there are really no ethical issues in using these cells. They're universally approved for use. Even, for example, the Catholic Church endorses adult human stem cells for therapeutic development, and they actually hold an annual adult stem cell congress on the Vatican campus yearly. So let's look at those embryonic versus adult stem cells. Embryonic stem cells here on the left side of the screen, again, are used for development of cells and tissues. And the embryonic stem cells primarily act through what we call differentiation. That is, the embryonic stem cells can transform themselves into other cell types. This happens through genetic and epigenetic and developmental cues in our body, but it is the embryonic stem cells that, through a bunch of signaling processes, can turn into any cell in our body. Whereas adult stem cells, they are located, and showing a picture of them on the right side of the screen. They're there in all of our tissues for maintenance and healing of cells in the tissues. Unlike the embryonic stem cells that primarily act through differentiation, adult stem cells primarily act through secretion of molecules, the many different types of proteins, for example, that they secrete, as well as secreting exosomes. Now, exosomes are nanospheres that package the different types of molecules and enable the molecules to penetrate barriers, to target tissues in need of help. So the molecules packaged into exosomes are very important. So again, the adult stem cells are acting primarily by the molecules they release. Embryonic stem cells acting primarily by transforming themselves into other cell types. Let's go into a little bit about stem cell science, the discoveries that science has made over the decades. How did we arrive to where we are today, where we can use the stem cell release molecules for skincare? Beginning in the 1970s, there was really the seminal work on stem cells, and in particular as it relates to skin. Howard Green at MIT, for the first time, showed that human keratinocytes, now most of you know that keratinocytes are a cell type in the epidermis, right on the basal layer of the epidermis, discovered that keratinocytes are a stem cell type, that they could be cultured, and that in culturing these keratinocytes, you could cause them to proliferate. So one cell would become two cells become four, and so on. That you could proliferate these keratinocytes. Indeed, they were a stem cell under the right conditions. During these times also, a lot of scientists here in the US as well as in Europe and in Japan were discovering that tissue could regenerate itself. And we were learning that stem cells were really the key underlying factor to much of this regeneration. So that scientists, man was learning to do what Mother Nature had been doing for us throughout our evolution. And then in the 1990s, while stem cell science was in its infancy, and stem cell science really is still in its infancy. every day we learn so much more about stem cells. But in the 90s, the first stem cell-based skincare product was initially researched, formulated, and developed for the marketplace. That product, using stem cell science, was created here in San Diego, California, where I am, and was used to treat wounded skin. Then in the early 2000s, the stem cell molecules released from skin stem cells were first put into a topical skincare serum, and that entered the market and was really a game changer. Then in the mid-2000s, and really crystallizing in around 2008, I put forth the concept of systems therapeutics, using systems therapeutics to treat both neurodegeneration as well as skin afflictions. And the systems therapeutic is really all about using multiple molecules from the stem cell types to treat a condition. Previously to the concept of a systems therapeutic, we were using one molecule to target one pathway to treat a condition. But we learned that the way Mother Nature was regenerating skin was using a collection of molecules, many molecules, released from the stem cell types to do the maintenance and healing. So this was, again, another real game changer to therapeutic development. Important to all of this was realizing that if you injected or somehow transplanted a stem cell into the body as a therapeutic, whether it was the brain, the skin, or elsewhere, what we were learning, many labs around the world were beginning to learn, is that stem cell molecules released from the stem cells were responsible for 80% and more of the healing benefits of the transplanted stem cells. This is a game changer. So instead of using the stem cell transplanted into the patient, we could, in the laboratory, create the molecules from the stem cells and then use those molecules directly to treat the patient. That's a distinct advantage for many reasons, why I won't go into here, but using the stem cells to treat the patient had many problems. You didn't know where the stem cells were going. You didn't know if they were alive. If they were alive, you didn't know if they were really releasing the molecules. And then also, I have a big paper coming out on this next month. When you transplant the stem cells into the patient, the stem cells themselves cause many problems. So you have to weigh the benefit to the harm that the stem cells are doing. In many instances, you don't want to transplant the stem cell, but the molecules don't have those same problems. So how is it we derive these molecules? And then once we have them, how is it we use the stem cell release molecules to treat the skin? We and a number of others showed, beginning in the 2000s, and kind of again crystallized around 2013, that the molecules were packaged into that nanosphere called the exosome. And the exosome was critical to the delivery of these molecules for treating a condition. These exosomes would allow us to break through barriers and target these molecules to particular tissues that needed to be healed. So again, that was a big game changer. So important to stem cell therapeutics and using the molecules is the identification of the stem cell types that are involved in the particular tissue to be healed. And you have to select out those identified stem cells. Then you have to process those identified selected stem cells in the laboratory. Take great care in the processing of the stem cells so that they will indeed in the laboratory release the molecules that you want. So again, if we're treating the skin, we want to use stem cell types that are derived from the skin. They're evolved to work in the skin and treat the skin, whereas stem cell types, for example, from bone marrow, are not normally present in the skin. And bone marrow stem cells are only in the skin during times of very significant wounds and infection. And so the bone marrow stem cells turn processes on in the skin that you wouldn't want to have happening day by day. They turn on high rates of proliferation, which can induce aging of the skin, can induce this cancerous state. The proliferative effects can be involved with fibrosis and scarring. And the bone marrow stem cells can induce a lot of inflammation. And chronic inflammation is destructive to the skin. Whereas the stem cell types resident in the skin don't have these negative consequences. In particular, it was discovered that the stem cell types in the skin that are critical to skin maintenance and healing are adipose-derived mesenchymal stem cells, as well as fibroblasts, and not the bone marrow stem cells. This was critical to developing molecules that you could use in a product to be used on a daily basis for skincare maintenance and healing. So to do this correctly, you want to take these adipose-derived mesenchymal stem cells and the fibroblast, control them, stimulate them, what we call in vitro, in the test tube, in a sterile environment. So if you do this with great care, you can culture these cells, stimulate them without antibiotics. So it's a very clean, sterile process. And then the molecules from the stem cells and the fibroblast are collected in the laboratory for formulation to be used in a topical skincare product. And interestingly, because these molecules from the stem cells are packaged into these exosomes, which I'll describe in a minute, these molecules are protected. And the molecules are protected from pH, from heat, from a lot of different issues. Therefore, the molecules can be packaged into a topical skincare product that can be used months later. Now, again, these exosomes that are released from the stem cells are critical. They are naturally produced liposome-like constructs. So I think many of you know about liposomes. So those are artificially made nanospheres. Typically, the liposomes may be 100 to 500 nanometers in diameter, whereas the exosomes released from the stem cells are typically about 80 nanometers in diameter. And packaged in the exosome are these various types of proteins, which I'll describe in a minute, as well as antioxidants and other important molecules to maintain and heal your skin. And also critical to the exosome, unlike the liposome, the exosome has proteins and polysaccharides tethered on the outside of the exosome that impart important targeting qualities to the exosome. So when you put it onto the skin, it can penetrate the barriers and really also know where to go to find cells in need of reparation. So what we call a smart liposome, that is the exosome, naturally made by the adult stem cells, secreted and packaged into the topical skincare products. So I mentioned to you the systems therapeutic approach. When we look at the skin, and really any other tissue, there are multiple types of adult stem cells resident in the skin. And when a tissue is damaged in need of repair, that damaged tissue will send out damage signals that will activate the stem cells and fibroblasts in the skin. And the stem cells and the fibroblasts in the skin, once they see these damage signals, can actually crawl around a little bit. They'll move. And they'll move not into the blood, but they move along the matrix, the extracellular matrix. It's kind of a pseudopodia event where they just crawl towards the area in need of repair. And then, once stimulated and in place, the stem cells, the different types, will release the molecules into that damaged tissue to institute the repair process, which includes the dampening of inflammation, reconstruction of the matrix, floating in antioxidants, and I'll describe this here in a minute, to really do a multiple attack, if you will, on the skin to bring the skin back to its normal place. So one of the things that the stem cells in the skin and in other tissues will do is to rebuild that beautiful extracellular matrix in the skin. And extracellular matrix exists throughout the body. So even in the nervous system and in the eye, extracellular matrix is critical for allowing damaged cells to be able to communicate with surrounding stem cells to seek out help in the repair of that damaged cell. And one of the things that stem cells will do when they receive a damage signal is to actually develop what we call a tunneling nanotube. So the tunneling nanotube is a physical nanometer construct that protrudes from the stem cell over to the damaged cell and develops a physical bridge between the damaged cell and the stem cell. And then over that tunneling nanotube, that bridge, molecules, the exosomes, will flow from the stem cell to the damaged cell to bring repair molecules to the damaged cell. Not only that, but just excitingly, something very new in the last few years, what laboratories are showing is that even organelles, mitochondria, so you all know about mitochondria, powerhouses of the cells, mitochondria from the stem cell can be transported along the tunneling nanotube to the damaged cell. So what happens in damaged cells a lot of times is that you lose mitochondrial function, and then you lose the energetics of the cell, and eventually the cell will go into a programmed cell death. So the stem cells can prevent all of that from happening. So again, the multiple molecules from the stem cells can move from the stem cell to the damaged cell, including over these tunneling nanotubes, to bring growth factors, bring antioxidant proteins, to bring proteins that clean up damaged proteins in the cell to be repaired. So there's a lot of different types of proteins and other molecules that go from the stem cell to the damaged cell. And that's what we use in the so-called S²RM technology, the stem cell release molecules, SRM, stem cell release molecules. And we say S²RM because we're using stem cell release molecules from 2 or more types of stem cells to institute repair, so S²RM. So for the development of skincare, stem cell technology and the molecules from the stem cells were really first used widely to treat burn conditions, and then a wide range of chronic issues such as eczema, psoriasis, and different forms of dermatitis, including atopic dermatitis. We're now using the so-called S²RM technology to not only treat, to remediate radiation burns in oncology patients that have been irradiated, but now we're showing that if we put the S²RM molecules on the patient before the irradiation, we can prevent radiation dermatitis. So that's very exciting for us. So the S²RM technology creates skin homeostasis. So what we're doing when we apply the molecules to the skin, we're bringing back the state of the skin to a condition when it was young and healthy. By putting all the normal proteins and other molecules back into damaged or aged skin, we're returning the skin to a normal state. And that allows us to treat a wide variety of conditions, including aged skin, acne, where you have inflammation, where you have different proteins out of the correct ratios, rosacea, and different sensitivities can all be treated with the stem cell release molecules. So these new products have been shown to be very safe over the years, to effectively create many conditions. And with these stem cell release molecules, we're treating a lot of what's going on in the dermis. We're treating what's going on also a little bit in the epidermis, particularly the proximal epidermis, where the keratinocytes reside. And we find that if we use, in addition to the stem cell release molecules, also a new barrier cream, which brings in the fatty acids needed to create the outer layer of the epidermis, that barrier cream works to really treat the stratum corneum of the epidermis. And then if you use the S²RM to treat the dermis, you have a beautiful combined synergistic effect to really recreate, to a large degree, normal skin in a lot of different conditions, including just simply aged skin. So let me show you some of the effects of using just simple topical application of these stem cell release molecules. So you can see here for aged skin, just simple topical application for 34 days of the S²RM technology can really rebuild the skin. What we're really looking at here is the underlying dermal layer and the extracellular matrix being rebuilt to a large degree. So wonderful effects with just topical application of the molecules with no other additional treatment. These photos are just untouched, natural photos showing the effects in 34 days. Crepey skin, we see a lot of that out there in the marketplace. A lot of you are treating crepey skin. And again, this was a 72-year-old patient. Simple topical application of the S²RM technology really returns that skin to a more normal, healthy state. Because you're renormalizing the skin, making the skin healthy, and rebuilding, amongst other things, that extracellular matrix, that skin, as a consequence, is going to look much more normal. For acne, we have similar effects. So the S²RM technology contains not only proteins, but it contains molecules, including certain types of proteins that quell inflammation, as well as other proteins and peptides that quell infection and quell the infection without turning on the induction of nasty cytotoxic chemicals, so that you have a nice effect, which is topical application of remediating the effects of acne. So again, here, this is simple topical application, the S²RM technology, no other treatments, and these photos have not been retouched. So in 7 weeks, topical application, the stem cell release molecules, dramatic effects in quelling acneic skin. For laser treatments, we see similar effects. So on the left, you can see a patient after a laser treatment, and then simply 5 days later, that skin is greatly improved. And had the skin not been treated with the S²RM technology, that skin would have remained very red during that time duration. We can treat conditions like rosacea. So again, simple topical application of the stem cell release molecules contain those molecules that will rebalance the skin, quell inflammation, and rebuild the normal matrix of the skin. Bring in antioxidants and a lot of other molecules, again, to renormalize that chronic condition. Now, for problems such as scarring, this lady, Brenda, was treated with a Fraxel laser at a clinic here in San Diego. And she has really beautiful but sensitive skin. And the Fraxel laser, unfortunately, had too much power during the treatment. And you can see there on the left, the Fraxel laser induced a scar. And that scar was present for 1 year when I met Brenda. And even though she had very good treatment in the best clinics here in San Diego, that scar would not go away. But when I was introduced to Brenda by a friend, I simply gave her the S²RM technology. And she used that for about a month, once or twice daily. And as you can see on the right, that scar went away. Again, simple topical application, no other treatments, no other products. The photo is not retouched. And for this thin scar, the scar nearly went away. If you look very closely at her skin, you can see just a slight, and you have to look very hard to see it, but a little ghost of what was once a scar in her skin. So in making the stem cell release molecules to treat these conditions, you have to be very careful about how you derive the molecules. So some technologies will crush the stem cells, and it's a physical and chemical crushing and really destructive to the molecules. Can create really molecular errors in the proteins so they don't function normally. Can have actually negative consequences when applied to the skin. It is a faster method and a lower cost method to derive the proteins and other molecules from the stem cells. But if you do it the way a number of us do it out there, and you allow the molecules to release from the stem cells, then the stem cells maintain their normal architecture. They're not hurt in some way and misfolded. And if you allow the molecules to release from the stem cells, they're packaged into those exosomes. So the exosomes not only protect the molecules, but give the molecules penetration and targeting qualities to those cells that are in trouble and need to have the cells or the molecules delivered to the cells. So again, with the crushing, you don't have the exosomes produced, and you have the proteins that may be misfolded. When you do the stem cell release instead of the stem cell crush, it's more time consuming, it's more expensive, but it delivers a very safe and efficacious product to be used for skincare. So there's, as I said, many different types of stem cells being used for skincare. Plant stem cells are used. Oftentimes what people think is a plant stem cell really isn't a stem cell at all. If they are using plant stem cells, the plant stem cells don't release most of the molecules needed to renormalize human skin. They will release a few of the molecules the skin needs. So the plant stem cells are not effective, are very effective. They'll do a little bit. Again, animal stem cells are sometimes used, but they're not optimal for treating human skin. Sometimes bone marrow stem cells are used to formulate the skincare products. Again, the bone marrow stem cells are only resident in the skin during times of major wounding and high levels of infection. And as a result, bone marrow stem cells will try to close the wound very quickly. They induce too much proliferation, which can induce scarring, can induce a cancer-like state in the skin, can induce too much inflammation that fights the infection. So they're really a suboptimal cell type to be using for skincare therapeutics. Whereas if you look at the stem cells that evolve to heal and maintain our skin, those are the fibroblast and the mesenchymal stem cell types derived both in the dermis as well as in the fat pad underneath the skin. Those are the stem cell types that you want to use in the skin. Now, this is true in other tissues as well. When you use bone marrow stem cells for therapeutics in other parts of the body, you have a lot of negative consequences. Research in the last few years has shown, because bone marrow is a place where stem cells and other cells recycle, so they come in from various tissues of the body through the blood and go back into the bone marrow, and you can actually have cancer cells go into the bone marrow, so that you may have cancer cells in the bone marrow from which you're using donated tissue to make your skin therapeutic. And if you're using cancer cells to derive your molecules, then the signaling molecules from the cancer cells can be present in the stem cell therapeutic. So that's been demonstrated. I have a paper coming out about that next month. So you have to be very careful about the types of adult stem cells that you use for your skincare therapeutic development. This was a very quick introduction to the use of stem cells and the science behind the use of stem cells for skincare therapeutics. And if you have any questions or would like more information, or perhaps some of the peer-reviewed PubMed listed publications that I have about this science, technology, and skincare, please do contact me. You can contact me through info@neogenesis.com or call into the NeoGenesis office, and they'll be happy to direct you to me and I can answer your questions by phone. Thank you very much for your attendance. Bye-bye.